Hello and welcome back. This is our last installment for our Chapter 3 Cell Structure and Function lecture. The last two topics I'm going to cover are endosymbiotic theory and just comparing some characteristics of everything we just learned about prokaryotes, bacteria, to prokaryotes, archaea, and take a look at those differences. So first, we're going to take a look at endosymbiotic theory. This was a theory proposed by Lynn Margulies back in the 60s-ish, 70s-ish, 1960s, 70s. Got to put the century in there because we were talking about the 1860s in chapter 1. Anyway, so she was making some observations along with other scientists as well. She just kind of culminated them together into this idea, is that there were quite a few similarities between the organelles of the mitochondria and chloroplasts of eukaryotic cells and just the characteristics of bacterial cells in general. So some of the key evidences, we'll take a look at those, are that chloroplasts and mitochondria, I'm just going to kind of abbreviate them, C and M, they both are about this similar size. They have similar sizes, about 2 to 5 microns, which is about the size of a typical bacterial cell. That's about the size of a chloroplast and mitochondria. Chloroplasts and mitochondria are double membraned. Now, other organelles, like a lysosome or peroxisome, only have a single phospholipid bilayer, where mitochondria and chloroplasts are double membrane. So if you remember, the mitochondria has the outer membrane, and then it has the inner membrane that's all wrinkled like this. And the chloroplast has the outer membrane, and then its inner membrane has been folded into all of these little things called thylakoids. And they're connected here and there. So they both have a similar structural pattern. Chloroplasts and mitochondria have small pieces of circular DNA. Right? So they have, like a prokaryote, has a small circular piece of DNA. So do chloroplasts and mitochondria, which is separate from the nuclear DNA of the nucleus of a eukaryotic cell. Chloroplasts and mitochondria also contain 70 S ribosomes within their structure. So they have basically all the makings of a prokaryotic cell. They have a membrane, they have DNA, they have ribosomes, and they're about the same size. So the culmination of those observations became the Indocin Bag Theory. The idea was, and this is a picture just I found off the internet because there's no picture in your textbook, that we have an ancestral prokaryotic cell. It's got the DNA, but it's not bounded by a nucleus. Then we have, as time progresses, maybe the nuclear envelope forming as a membrane just kind of folding in on itself. But then after, some interactions with some maybe other critters, it might have engulfed an aerobic heterotrophic prokaryote. Now, we haven't talked about what a heterotroph is, but a heterotroph is an organism that consumes nutrients to burn energy. So as opposed to an autotroph, which is like plants, they use the sun's energy to make food and burn. So the idea goes that these ancient kind of primordial cells engulfed their neighbors, in this case, a heterotrophic bacteria, but instead of breaking it down and eating it, it just was like, hey, I'll let you stay inside of my cell if you give me some of your energy. So they created a symbiotic relationship, a mutually symbiotic, a mutually beneficial relationship where the heterotrophic bacteria will share the ATP that it makes by its metabolic pathways, and the engulfing cell will not destroy it and will give it nutrients and a safe place to stay. So these developed into our mitochondria, and then that cell line went to produce like animals and fungi and some protists. Where another branch off of, after that event is that they engulfed a photosynthetic prokaryote, and same type of idea. Instead of consuming it and getting rid of it, it allowed it to stay. Not that it gave permission, but this, evolutionary ideas, engulfed the photosynthetic prokaryote to produce the ancestral plant and other photosynthetic organism cell lines. So this is how the chloroplast came about. So this idea that way back in evolutionary history, it wasn't just the development of these organelles within eukaryotes themselves, but the incorporation of existing prokaryotes into each other set the stage for the development of what we understand as modern day eukaryotic cells with these membrane-bound organelles. And the inner membrane, right? So if we're going to take a look at the mitochondria, this inner membrane would have been the plasma membrane of the original prokaryote, and the outer membrane would be the phagocytic vesicle that did not get digested and was maintained for both chloroplasts and mitochondria. So it's a really cool concept, and it was not widely accepted, and it's still argued today. But in general, there's always, paradigm shifting challenges to certain things. Lynn Margulies' idea is pretty much widely accepted as how modern day eukaryotic cells have come to be. Now, there's some things that can't be explained by this, like the double membrane around a nucleus. We didn't engulf the nucleus, so why does it have an inner and an outer nuclear envelope? It also does not explain the genes on the circular DNA in the chloroplast and mitochondria don't code for the enzymes needed inside of those. Those genes are in the nuclear DNA. So there's still some things that haven't been figured out, but there is quite a bit of evidence to support this idea of the endosymbiotic theory. Okay. All right, so let's take a look at some differences between archaea and bacteria. So again, archaea and bacteria are both prokaryotes, single-celled organisms. They lack a nucleus and membrane-bound organelles. They still fall under the category of prokaryotes, but there's enough differences between these single-celled organisms, archaea and bacteria, that they actually have their own domain. So there's eukarya, which covers all of the eukaryotic organisms, bacteria, and archaea. So we just spent most of chapter 3 talking about bacterial characteristics. Now we're going to take a look at archaeal characteristics. So as far as glycocalyces is concerned, there's some similarities. They can make biofilms. As far as flagella are concerned, archaea flagella rotate a little bit slower. They're a little bit thinner, about half the thickness of a bacterial flagella. They have different amino acid sequences in the proteins in the flagella part, the whip tail, and they are powered by ATP, where bacterial flagella, their power source is concentration gradients like hydrogen. Hydrogen flowing from a high to low concentration is what drives the spinning of the flagella, whereas in archaea, it is spun like eukaryotic flagella, which is powered by ATP. So there's similarities between archaea and eukaryotes, actually. They have fimbriae and hammy instead of fimbriae and pili, and they're similar in structure and function. The membranes, archaea plasma membranes lack phosphate groups on the lipids, and they have stronger hydrocarbon linkages within their membrane structure, which might account for some of the environments that these archaea live in. These extremophiles we'll see in chapter 11. I mentioned this before, the cell wall contains no peptidoglycan, so they have other structures to make up their cell wall, but peptidoglycan is only found in bacterial prokaryotes. And then they also have 70S ribosomes, but they are more similar to the 70S ribosomes that are found in our eukaryotic mitochondria and chloroplasts. So the idea is, again, looking at evolutionary history, if we have a common ancestor that branched off into eukaryotic cells and prokaryotic cells, the archaea branched off here. A. So the idea is archaea are more closely related to eukaryotes than they are to prokaryotes. So it's kind of interesting. They're finding more of those more similar connections between archaea and eukarya than they are bacteria, I should say, bacteria and archaea, even though these are both prokaryotes. Okay. All right, so that wraps up our chapter 3. We did it. Lots of little mini lessons covering those topics. Send questions, email, stop by an office hour if you need more clarification. All right, I will see you next time.